Study guide. If anyone needs a copy of the study guide, have it. Take two if you want. You're extra. What I will do on Wednesday, I forgot.
I'll take another one.
I was thinking of doing. Anyone else?
Sure, I'll take another one.
You take another. Take another. Got all these extras.
Just in case I can't find one.
What I was planning on doing today, and tell you the truth, I forgot. I knew there was something I didn't do for today. I'll give it to you Wednesday, and that would be some. Or I'll put it on my CR. I'll do it tonight, so it'll be on my CR under what? I'll put it under resources, I guess, and there'll be some sample study questions. They'll be similar, very similar to the questions will be on the exam. And I'll print it out and give it to you on Wednesday too. But I'll do that tonight on my CR. So very similar kinds of questions. You want one of these or questions? Yeah. Go ahead. A little louder. I'm sorry.
If you don't get through the lecture today, will you just continue to Wednesday?
Oh, okay. Well, to be honest, what I was going to say, I'm glad you brought it up, and I think I will get through. But technically the exam was going to go through this Wednesday's lecture. But considering that, last exam we didn't get as far as I wanted to, and so we're actually starting with cell respiration. It wasn't on the exam, so I figured this would be a good, handy place to end. But if I don't finish it, then yes, I would like to carry it through. Right, because I really wanted to finish this topic. I think I'll finish it. I hope, anyway. And by the way, I think this stuff is interesting. I don't know if you guys, how you feel about it, but we will be talking starting next time about like some human genetics, and it gets kind of fascinating, and just to see some of the things that can go wrong. It's fascinating. It's not good. It's terrible. Things that go wrong are terrible. I'm not saying it's good, but it is fascinating.
They made one. A genome. They made a genome that works. I just read they made an artificial genome that did not destroy itself.
Oh.
Cell is actually replicating itself.
I'll have to check it out. You have to give me the source.
Alright, it's awesome. I will. I'll send it to you.
Okay, so questions before I start.
Yeah.
Yeah.
Okay, we're going over the study guide.
Study guide. Good.
We're going over the study guide, and there's a question about. Okay. Study guide. Why was it uncertain until the 1940s and 50s that genetic materials DNA reduction proteins? I have it written down.
Wait, so where are you? The question was why? Oh, why was it uncertain?
I don't have that in my notes. It says that there were studies, but it doesn't say which ones.
Right. Basically, it was this. First of all, they knew about proteins. They knew how many functions proteins had, so they figured it must be proteins. Besides, proteins are made of 20 different amino acids, so you can have such a huge variety of possible structures. And they knew about nucleic acids, but they're only made of 4 different nucleotides. So they figured, how could you take something with only 4 different nucleotides and have it be able to do whatever you need to do to code for life? Because they didn't know how it coded for life in those days. They just figured there wasn't enough information content. If you only have 4 different bases. Once they got that structure of DNA, and they saw that there was this definite base pairing pattern, and then they started saying, hey, okay, now we know how it can duplicate itself exactly, and then, hey, how can it be used to run the cell? Well, how does the cell run anyway? Proteins, enzymes, And so they figured, okay, how could DNA be that? And it became a problem in coding. And they hypothesized a lot of things about the genetic code and all that. And remember, I've said so many times, if you can ask the right question, you can probably get the answer. But by being able to ask these questions, they could say, hey, how could we do it? And they said, oh, it's got to be a 3-base code. Well, if it becomes a 3-base code, then you have enough information, But it was just, they didn't know enough about DNA yet to see it. Anyway, that's the idea. The main thing I'm trying to get you to think about is, that structure, getting that structure was critical to understanding how things work.
Who was it? I forget the name of the people. The 2 people that came up with that double helix.
Well, Watson and Crick. Those are the 2. Watson and Crick.
Okay, that's who.
Yeah.
Okay, that's fine.
Yeah. Yeah, no, they are the ones. And as I said, the model alone allowed people to ask all kinds of fascinating questions that they could then answer. Without the model, they weren't sure how to ask the questions. I mean, so anyway, it's just kind of that's the idea. I'm not going to ask you a question necessarily like that. It's really to get your thinking going. But I'll ask you questions about the model. Yeah.
Yeah, I have one more question.
Oh yeah.
A mutation.
A mutation.
What are some of the causes of mutation?
They're
I
random.
didn't get into that, did I? I think I didn't get into it. The simple, and by the way, you can define these things in different ways. One simple definition of a mutation is a change in DNA nucleotides that'll change something about the protein that's going to be coded for. And then actually, you can talk about a neutral mutation that doesn't change the codon or doesn't change the amino acid. But good question. I didn't get into it, but how can mutations occur? They can occur through radiation, cosmic rays, X-rays. That's why they say you want to limit your exposure to X-rays. If you need an X-ray, have an X-ray, but don't just overexpose yourself. So radiation, cosmic rays, we're constantly bombarded by cosmic rays. We can't stop that, so that happens. UV light, UV doesn't penetrate very far, but it can cause mutations in your skin cells because it will penetrate a little bit into your skin. So those can cause mutations. There are various chemicals that can cause mutations. They're called mutagens. Most mutagens can be carcinogens, meaning they can cause cancer. It's not an absolute, but most can. Then it can happen just randomly.
Yeah,
just total random. No one knows why. So. But again, keep in mind that mutations don't occur. Well, I don't want to say they don't occur often. They don't persist often because if it happens, first of all, if it happens just a regular old cell, it may not cause any problems. If it happens in a gamete, then it can be passed on to other generations. The mutation can. But the other thing about it is if your cells are just in S phase and duplicating all their chromosomes, and a mutation occurs, there are those proofreading enzymes that can cut out the bad part and replace it with the good part. So that's why mutations aren't common. They do occur, but does that answer your question? Or did it more than answer?
No, I answered it. Thank you.
All right, let me go back and review a couple of things about Mendelian genetics so we can go on to, we went through Mendel's first law, but I'll review it again, and then we're going to talk about Mendel's second law and do some dihybrid crosses. Then I'm going to talk about some variations on Mendelian genetics. I think this is kind of neat stuff. Because it starts to show you how variation, well, we have variation, but how it gets passed along, from generation to generation. And why, in a family you might have several siblings, but you all look different from each other, Anyway, let's go back. Remember we talked about, well, first of all, we have pairs of chromosomes, right? So eukaryotic cells have pairs of chromosomes. We've known this for a while, haven't you?
Mm-hmm.
So they have, that's what we have. They have pairs of each gene. Now, so we get two, for whatever it might be, if we just make it simple and say there are two genes for eye color, you inherit one from the mom, one from your dad. Okay, so you have a pair of genes for eye color. There's another word, I don't think I introduced this in lecture, but I want to introduce it now, and that's the word allele. If we use the word allele, so we'll say for a given pair of genes, and it could be, as I said, for maybe eye color, say, for a given pair of genes, if their is if there are different types, if there are two or more types of that gene, forms of the gene, not forms, what's a better word? Examples of that gene, I don't like the word example. For a given pair of genes, if there are two or more variations, that's what I want, variations of the gene, we use the word allele.
To describe the genome?
To describe the genes. No, well, those genes. Remember, the genome refers to all the genes. So here we're just talking about just one
specific,
pair of genes.
Yeah.
Just say the genes for eye color. We use the word allele to indicate this. So we can speak of the alleles for eye color. You can interchange the word gene and allele. That's perfectly okay. It's not like it's an absolute. So I can say, hey, there are two genes for eye color, or I can say there are two alleles for eye color. You can use them interchangeably. Preferably, if it's just the same gene that somebody has, you just say, hey, they have the same gene. But if there is variation, then we usually use the word allele. It's not an absolute. It's not a big deal, but you will run across this word all the time, so that's why I want to use it. So I'll say there are two or more alleles for eye color.
So meaning that there's one that's blue, one's brown, one's green.
Maybe one that's green, one that's exactly. So there may be several. It could be that you have more than one allele for eye color. Eye color is a little. We usually say it's simple and say there's a dominant brown and a recessive blue, but it's a little more complicated than that. So there are other things that do come into play. But we usually, a lot of times, they oversimplify, make it easier. Otherwise, it gets too complicated. So anyway, so we use the word allele. So then of course we talk about dominant and recessive. If one allele, so I'll go ahead and use the word allele now, and I used the word gene last lecture, but now we'll use the word allele. If one allele is expressed I'll say whether if there's one copy or two copies, whether the person, the individual has one or two copies of that allele, then we say that allele or gene, again you can say the word gene, is dominant. I'll use the word allele since that's the word we're using. That allele is dominant. If only expressed if present if two copies are present. Let's see. And the allele, we talked about this last time. The allele is recessive. So we have these are two important words: dominant and recessive. And again, two copies. We're talking about one copy from your mom, one copy from your dad. So if you only need to have one of them, one copy, it doesn't matter what the other copy is, but it's going to be expressed and it's dominant. But if you have to have two copies to be expressed, then we say it's recessive. Okay? We talked about this last time. We use the word homozygous and heterozygous. Remember that? homozygous and heterozygous. Homozygous means you have two of the same alleles. Heterozygous means you have two different alleles for some particular gene or a trait. Say for a trait, if there are two of the same alleles present, then the individual is homozygous for that allele. Leave a little bit of space here. If two different genes, two different alleles, and heterozygous. I'm just reviewing.
And in heterozygous, the dominant prevails.
The dominant will prevail. Yes, the dominant will be expressed. Absolutely. Okay. So again, these are just words that are very important words because they'll be used a lot. Now, the thing is, for a given, whatever the allele is, the person could be homozygous dominant or they could be homozygous recessive. I mean, you would have to say, okay, that person is homozygous dominant for eye color, say,
brown eyes.
Homozygous recessive. So I'll say, so could be homozygous dominant. This is always going to be expressed if you're working a problem. That's why I want to make sure you understand this. Or could be homozygous recessive. If you're heterozygous, that means you have one dominant and one recessive, right? You have a choice. You have one dominant and one recessive.
How can you be homozygous recessive then?
So how can you be homozygous recessive?
Yeah, what is an example?
Well, so let's take eye color. So we'll say, and again, it has to be given to you what's dominant and what's recessive. So and again, we're oversimplifying it. I think eye color is a little more complicated, but that's okay. We can make it simple. We'll say brown eyes are dominant blue eyes, or dominant to blue eyes. I said over, but you can say to. So we'll say brown eyes dominant over blue eyes. Now, when you're going to work a problem, and I will give you a problem on the exam, so that's why I want to practice these things. If you're going to work one of these, and in lab, you're going to do these things, same kind of thing. If you're going to work a problem, write all this information out. I mean, it'll be given, let's say, okay, brown eyes are dominant to blue eyes. Okay, good. But now what you want to do is give some letter. So remember, use capital letter for the dominant. Now, both brown and blue start with a B. Usually they'll take the first letter of whatever the dominant trait is. They both are B. So you say, okay, we'll say B for dominant, big B for dominant, and little b for recessive. Remember I mentioned last time, be careful for letters that, like an S and an S, or an S and an S, you could, if you're writing fast, you might be not sure whether you wrote a big or a small one. So you have to be kind of careful. But at least a big B and a small b, they look different. So we're going to use the letter B to indicate the dominant gene or allele, and the little one for the recessive allele. So now, write down your possible genotypes. This is the way you should work this. And then I'm getting to your answer, Rachel. I'm going to answer your question. But it's good because I want to do this first. Possible genotypes and phenotypes. So what are the possible genotypes that a person would have? You have to have two B genes. Person, you're going to inherit two B genes. You could inherit two big B's, big B, big B. You could inherit a big B and a little b, or you could inherit a little b and a little b.
So
Okay.
the first one's.
And if
homozygous.
brown is dominant, then it doesn't matter whether you have two big blue B's or a big little b, little b. Your phenotype will be brown eyes. Whereas two little b's would be blue eyes. All right. So two possible genotypes to give you the same phenotype with respect to that dominant. Now, what about the terms homozygous and heterozygous? This one right here, the two big B's would be homozygous dominant.
Because they're the same.
Okay. And the two little b's would be homozygous recessive. Can you see that, the red up there? Is it readable? I'm sorry, the red's not very good.
It'll be revealed.
Can you read it okay? You guys can read it okay? So the big B is homozygous. By the way, when you say it, don't just say BB because it gets confusing. Say big B, little b, whatever. Anyway, the two big B's would be homozygous dominant. The two little b's would be homozygous recessive. So that answers your question, right? But this is a way to set it up if you're going to do a problem. So here's the thing. So we're going to go back and review, the law of independent assortment, or the law of independent segregation. We're going to go back and review Mendel's first law. So I'm going to erase all this. Get your notes and stuff like that. I better erase it.
But it helps to note that homo means same and hetero means different.
Different. That's right. Exactly.
So would the big B and little b, would that be considered heterozygous?
Yes, so that's heterozygous. I didn't write that down. I meant to write that down. Yes, absolutely. That's a heterozygous state. And what they'll say in a problem is, okay, a person who's homozygous for brown eyes but heterozygous, heterozygous for brown eyes marries a person who is blue eyes or something, or homozygous recessive. I mean, they'll say those words. So that's why it's important to know the term, what the terms mean. So yes, let me put this one. This one then, of course, is heterozygous. So if a problem stated that a person was heterozygous for eye color, and you were given the fact that brown eyes are dominant over blue, then you're assuming, okay, if they're heterozygous for eye color, they're going to be brown eyes, and they're going to be a big B, little b. Automatically, you know that. Or if they're homozygous recessive for eye color, and you know that blue is recessive, there's two little b's. Yeah, I mean that's why the terms, you just get used to using the terms, so you use them over and over again. And you'll do this in the lab this week, so it'll make it nice and easy after enough practice. But here's the point. As far as solving any of these genetics problems, is you take a person or whatever the organism is, and I'll say person, but it could be a tree or whatever. But you're taking the individual, and you're going to mate that individual with another individual, and you'll know what the genotypes are. But you always have to remember, wait a minute, the person has two B genes, but when they make gametes, how many B genes go into a gamete? Only one, right? Gametes only have one of every chromosome. So they're going to have one of every gene. And that's what you have to remember, that when you're making gametes, they only get one of whatever the two genes are, or two alleles. Again, you can use allele and gene interchangeably, and I will use them interchangeably. But keep in mind that the gametes will only get one of those. So if I said that you were going to let's just say a person with brown eyes, let's say homozygous for brown, let's say person, we'll say homozygous brown eyes marries a person who is, I'll say person, that's supposed to be a person. Person one, homozygous brown eyes marries person two. And we'll say these are homozygous blue eyes. Okay. What is the genotype of each of these? We say, what's the genotype?
One is big B, big B.
Big B, big B. And this person is?
Little b, little b.
Little b, little b. And they're going to mate. What are the possible gametes that this person can make? Gametes.
If they only have big B, big B.
I'll say possible gametes. Possible gametes.
Big B,
They're all going to be, no, this person.
No, they're all going to be the same.
They're all big B. That's the only kind of gamete they can make.
Why?
Because he doesn't have any little b's to put in it, right? And this person
no little bit.
can only make little b. Gametes can only have one little b. Now, if these cross, what do all the offspring have?
They're going to have brown eyes.
F1 generation would all be big B, little b. Right? They'd all be big B, little b. Because they get a big B from one parent and a little b from the other parent. So what's that? That's their genotype. What's their phenotype? Brown eyes. But what if a person who's has this genotype, heterozygous, marries another person who's that same genotype? We won't cross them, sister and brother.
It'll be a 3-to-1 ratio. 75% of the children will have brown eyes.
But let's just say that some other family. Gotta be careful around here. Okay, I don't want to make value judgments. All right. So let's say if we cross an F1 with an F1, and by the way, these could be dogs. You have dogs that have brown eyes, so it could be dogs. And there you cross brothers and sisters, and you got uncles and grandfathers.
Oh yeah,
let's
dog
do an
breeding's
F1
rough.
Cross an F1, and let's say that these F1, this person is a big B, a little b, and this person is a big B, a little b. What are the possible gametes of this person? First person. Possible gametes.
Big B, big B.
This person right here could have a big B. Whoops, that's supposed to be a big B, sorry.
And a little B.
Or a little b. This person, same thing, right?
Yeah.
Big B or little b. Now, this is Mendel's first law, the law of independent segregation. That if you're taking somebody with some individual with now these are heterozygous, right? They're heterozygous variecolor.
Yeah.
When they make gametes, one of the big B will go into one gamete, the little b into another gamete. You won't get when this F1 person, this first person here makes a gamete, he doesn't put two B's in it. His gametes get one B gene. It could be a big B or it could be a little b. And you can figure out 50% will have a big B, 50% have a little b. Same thing with the second person. In the previous one over here, there was only one possibility of what gametes you get. All the gametes would get a big B. Half of them would get the one on the left and half would get the one on the right, but they're the same, so it doesn't matter, right? But and here the gametes, half of them are big B, half are little b. So when you go to say, what are the possible outcomes if you cross these people? You say, okay, well now here's where you can make that Punnett square. So remember this Punnett square we did? And this is a convenient way to do it. You can do a lot of these in your head. This one you can do in your head, but just good practice to do it with a Punnett square because we're going to do a dihybrid cross in just a minute. It's harder.
I realize this is probably further on.
Yeah.
But if brown eyes are considered like dominant traits, sometimes you consider recessive traits.
Right.
What would green eyes be considered?
Well, see, that's a good point, and that's why I say I'm not quite certain that it's only two genes, and there may be some other genes involved that will change things. So that's why
serious
I'm
blue
oversimplifying
recessives.
this. And you could probably look it up online. And see what they now propose for this, how many different genes there might be, or alleles there might be for eye color. But I'm just making it oversimplified on purpose so we can do a nice easy thing. Yeah.
Okay, so what happens when someone has two different color eyes, like a brown eye and a blue eye?
That's a
And
crazy.
that does happen occasionally. It's rare.
They explained that on X-Men First Class.
Well, let me just real quickly explain what happens. Early on in development, egg and the sperm fuse, you get a zygote, then it starts dividing and dividing and dividing and dividing. And ordinarily when all those divisions, those are mitotic divisions, so every cell should have the same genes, and so they should have the same genes, which would be for the two different eye colors, which should be giving whatever you should get. But every so often, in forming the iris, which is the colored part of the eye, there's certain cells in that dividing zygote, there's certain cells that are going to form the iris, and every so often, one of those genes, even though they should get both copies, only one goes to one cell and one goes to the other for whatever reason. So then they only have the one gene to express, so they do it. So it's unusual. It's an unusual thing. Calico cats are another different story, but I'll get into that not today. Maybe today when we get down, but not right now. But maybe we can get into the idea of calico cats and why they're always female.
I was gonna say they can't be male.
Almost always, but yes, almost always.
There's an almost.
Almost always. Anyway, we'll come back to that. And actually, I might get to it next lecture when we start talking about chromosomal abnormalities. But anyway, back to this. So let's do this. So now we're crossing this F1 cross F1, but in both cases they're heterozygotes. They're both heterozygotes. So what you do when you do these Punnett squares, and how many different genes how many different gametes can this person make?
Two.
This can make two. So we're going to make it a 2 by 2. So what you want to do is just make it 2 by 2 like this. And I usually leave a little bit extra, but so you want to have 4 boxes. So it's 2 by 2, 2 boxes by 2 boxes. And what you do is you put the gametes, the possible gametes. Now this is easy for this, but we're going to do the dihybrid in just a minute. Put the possible gametes along here and also coming down here. Now it doesn't matter, which one you put down, which one across. It doesn't matter a bit. But so let's just start with the first one. There's 2 possible gametes for this person, right? I'll put them across. One possible gamete could have a big B in it, and the other possible gamete would have a little b. For this guy, his gametes are going to come down the side. One possible gamete's a big B, one possible gamete's a little b. So those are the gametes. In the internal squares is what if a gamete with a big B met a gamete with a big B, say a sperm with a big B met an egg with a big B, what would the individual have? He'd have, and by the way, the offspring are going to have 2 B genes, one from each parent, 2 big Bs. If it was this way, it'd be a big B and a little b. Here would be a big B and a little b. And there would be 2 little b's.
And that's why there always is a 3-to-1 ratio.
So this actually gives you, in terms of phenotypes, 3-to-1 ratio. Because what are the phenotypes? Let's write the phenotypes. 2 big B's would be brown, right? Big B, little b, brown. Just dominant. Big B, little b, brown. And so only this one right here would be blue. So in terms of phenotypes, a phenotypic ratio. And be careful because, the phenotypic ratio is 3-to-1 brown eyes to blue. Or you can say 3 browns to 1 blue. That's the phenotypic ratio. But let's just say that, you look at one of these individuals with brown eyes. And I say, okay, here's an individual with brown eyes from this cross. That his phenotype is brown eyes. What's his genotype? Could you tell me?
Yeah.
Or could you be sure? Let's put it that way. No. You can't be sure. Could be 2 big B's, could be a big B, little b. You don't know. All you know is brown eyes. That's the phenotype. But you don't know whether he's the one who got the 2 big B's or the big B, little b. You don't know.
If you're looking at someone with like green eyes or blue eyes, you know for sure that they
Okay, if you look at the green we're saying we're not sure about right now. We're leaving that out of the equation just to make it easy. But if you see someone with blue eyes, you go, you know what they are. They're homozygous recessive. You know their genotype because that's the only possibility. Does that make sense? Yes. But with the dominant one, you don't know just by looking. You have to do some breeding experiments. So to be sure. But anyway, that's the idea. But again, back to the law of independent segregation. When gametes are made, now you have everyone has 2 copies of every gene, right? But when gametes are made, 1 copy of that gene or that allele, one allele goes to one gamete, one allele goes to the other gamete. Okay, because we remember that whole idea of meiosis was to reduce the number of chromosomes in half. So instead of having 2 chromosomes in the gamete, you have only 1 of each chromosome in the gamete, which means 1 of each gene on whatever that chromosome is. Sinking in?
Yes.
You sure?
It's mostly review, so yeah.
Well, this is what we did last time, and this is fine. I want it to be reviewed because now I want to go on. Yeah.
So Ralph, again, the gametes you only get 1 copy of the gene per chromosome.
Yeah, for every because you only but let's say it the other way. A gamete has 1 of each chromosome, and so therefore only 1 of every. Say chromosome number 1, 1 of every gene on chromosome number 1, 1 of every gene in chromosome number 2, whatever it might be.
So 23.
And then when that when the egg and the sperm meet, then of course they'll have
46.
pairs of chromosomes again, 2 of every one. But the trick when you're doing these Punnett squares to figure out probabilities is gametes, possible gametes. You've got to remember only 1 of the B genes is going to go into the gamete. And now this person has 2 different alleles for that. With in this case, eye color. He's got 2 different alleles, but half his gametes will have the big B, half will have the little b. Okay. With this person, like they take this person down here. If that person is going to have some offspring, what about all of his gametes? Every one of his gametes will have a little b. So all his gametes are little b.
Huh?
Right? For this guy.
Yeah.
And all his gametes are going to be big B. But these heterozygotes, half will be big B, half will be little b. That's why it's important that you use these the words heterozygote versus homozygote, or heterozygous and homozygous. Okay. Let's look at the dihybrid cross. Okay, let's do the dihybrid cross. And you're going to do this in lab. Actually, it was kind of fun because there's this stuff called the genetic corn, and it's bred especially so you can do these experiments. And it's also very pretty. But you have corn that can have in this one corn, it'll have purple or yellow kernels. All so if you look at it, a whole bunch of purple kernels, a whole bunch of yellow kernels, all mixed up. And some of the kernels are smooth and some are wrinkled. And it so happens that the purple is dominant over yellow, and the smooth is dominant over wrinkled. And so then you're going to go through and actually count them to see the ratio that you get. And we're going to do that I won't use the purple corn. I'll use one of the ones from maybe Mendel, but you'll see how you get the ratio when you're using 2 different genes. 2 different traits. I should say 2 different traits. So let me make sense out of this. So let's look at seed color. Say what Mendel was doing. Seed color and what was I doing?
That was the smoothness.
We did pod color, we did seed color, we did smooth and wrinkled. Let's see, what do I have? I was going to remind myself of which one. It doesn't matter which one we do. Let's do here. The ones that I have, yellow. Let's see. Okay, here we go. Let's do some tea plants. Tea plants. Okay, we have the seed. Let's do color. Seed color. Seed color. Yellow. Dominant.
Green recessive.
Over green. And seed shape. Oops. This is what I did last time, I think. Seed shape was smooth.
Over wrinkled.
Dominant. Over wrinkled. Okay, so when Mendel was doing this, he was originally he looked at things were just yellow seeds and green seeds, and the ones with yellow seeds would breed true, and the ones with green seeds would breed true, and then when he crossed them, he found out which was dominant and which one was recessive. And then he did the same thing with seed shape. He found the smooth was dominant over over wrinkled. But then, yeah, go ahead.
So when you say dominant over green, you would say green is recessive every time?
Yes. Yeah. And I could have said yellow dominant, green recessive, exactly. And smooth dominant, wrinkled recessive. Yes, it's perfectly fine to say it that way. Same thing. Let's get some. Just give our letters just to make it easy. We'll use what we did last time. We'll say okay. We'll use big S for dominant, and little s. I'm making it different from. I'm writing it differently
For
recessive.
recognition
Just make it
purposes.
easy, okay? And we'll say big. Oh dear, we have smooth and wrinkled. Oh, that was not. That was down here. What am I doing? What am I doing? Sorry.
We could do C for color.
Yeah. Well, I was going to do Y, big Y for dominant. I'm sorry, I was doing C color. I was doing the other one first. And little y for green. Jeez, big Y dominant, little y recessive. We'll do it that way. Okay. And big S for the dominant. And little s for recessive. Okay, so start with that. Now let's talk about possible gametes that will give you, yellows and greens for seed color. So gametes, we'll say possible gametes and phenotypes. So you could have big Y, or you could have big Y, little y, right? Or you could have little y, little y. Three possible gametes, for not gametes, sorry, I said possible gametes, sorry. Possible genotypes. God, let me just, I goofed. I don't want to goof. Let me make sure we have these words right. Genotype is the genes that are present. And phenotype is how the genes are expressed. I didn't mean to talk about gametes there because we're not ready for the crossing yet. My mistake. So possible genotypes that a pea plant could have. Could have two big Y's, a big Y, little y, or two little y's. What would be the phenotype of two big Y's? Yellow, right? And the two little y's would be green. And notice this was different from pod color that I did last time. Pod color, the green was dominant over yellow, but apparently in the seed color, yellow is dominant over green. What about for the smooth and wrinkled? We could have two big S's.
Two big S's, big S,
and a
little
little
s, and two spots.
And again, the two big S's, if you have a big S present, you're going to be
Smooth.
smooth. And if you have two little S's, you will be
Wrinkled.
wrinkled. Now you're probably going to ask, is that true for human skin? As you get older, does your genotype change? Your skin starts getting... No, it doesn't happen. I'm just kidding. Okay. All right. So I was just teasing. Skin does lose elasticity. I can tell you that for sure. Skin just doesn't do anything. It's gone after a while. Anyway, and gets very thin. But whatever. We're not talking about that. We're talking about pea plants right now. Okay, so Mendel had established, that yellow seeds were dominant over green seeds, and smooth seeds were dominant over wrinkled seeds. Now, and because he was just looking at monohybrid cross, just doing a one seed color cross with a different seed color. You cross a yellow seeded plant with a green seeded plant. And he done the same thing. Smooth seeded plant with a wrinkled seeded plant. Now he said, let's take one that's yellow and smooth, or yellow and wrinkled, or green and smooth and green and wrinkled. Let's cross these together and see what happens. But what he did, he took some true breeding ones first. So he took, so Mendel, we're going to do what's called a dihybrid cross.
Mm.
Okay, this is, we didn't get to this last time. This is new stuff. That was all review, but it's important review, and I wanted to do it. But now we're going to do the dihybrid cross. This is new stuff, okay? This is going to be, we're leading into Mendel's second law. So if you want to cross true breeding plants, and you know, these are these peas that self-pollinated, so he could get the true breeding kind, and he would cross, say, a yellow smooth, yellow seed, and smooth seed. This is one plant. Cross with a green seed and a wrinkled seed. I know I could say yellow smooth and green wrinkled, but I did it in separate ones. So he crossed those. Now those are true breeding. If they were true breeding, what would be the genotypes? So let's put the genotypes.
So that would be big Y, big S.
Big Y, big S, cross with
small y, small y.
Little y, little s, right?
Yes.
Okay, because these were true breeding, so they were homozygous for both traits. In the case of the first one, it's homozygous dominant for both traits. In the case of the second, homozygous recessive for both traits. Okay, that makes sense? I'll write that up. So this homozygous dominant for both traits. And this other one was homozygous recessive. Now, again, he wasn't sure what was going to happen when he tried the experiment, but it turned out when he did this, all the offspring they had yellow seeds, yellow smooth seeds. Okay, so this would be the parental. This would be the parental P generation. The F1 generation, when you cross them, all were yellow smooth. The dominant. But then when you cross these, you got all kinds of combinations. But let's just deal with what the gametes were. What were the gametes? What are the gametes that were formed from this one? Let me see if I can do this. Can I speak this in? No, let's do this. So all were yellow smooth. Let's do the gametes. So the gametes from this. Gametes from this guy. What are the gametes from this first guy?
One big Y.
They're always going to be one big Y,
One big S.
one big S.
But it has two?
But gametes are divided.
Well, but it's going to have one Y and one S. But this guy, they're all going to be big Y because there's no choice, and all big S because there's no choice. So all the gametes will have a big Y and a big S.
And
right?
that's because there's doubles.
Because we're looking at two different traits, two different genes, and they're on separate chromosomes. Okay. And whereas this one, they're all going to be
Little Y and little S.
little y and little s. Now, these are the gametes. They only have one y and one s. Now, when you cross these, this is the parental generation, right? The F1, what's it going to end up? They're all going to be, will be big Y, big S, little s. Now, does that make them yellow smooth?
Yeah.
Yes. Okay, they're all yellow smooth. Okay, so that's fine. This is fine. This is what he did. But now, because Mendel was a good scientist, he got one set of experiments. What can we do now? Let's cross those F1s with F1s and see what we get. So you okay so far?
Mm-hmm.
Okay, that's straightforward. So now he said, let's cross the F1s with F1s. Again, same, we're using the same, everything's the same there. So now we'll cross
I already see how this works.
F1 with an F1. And by the way, with plants, you can always go back and cross an F1 with the parent. You know, you can do all these things. But anyway, so let's write down what we're crossing. We're crossing, so the genotype of the ones we're crossing, genotypes. Okay, we're talking about they're all this. This is the genotype for both of them, right?
Mm-hmm.
Heterozygotes. So big Y, big S, little s, cross with a big Y, little y, big S, little s. All of them are that way.
Mm-hmm.
Now here's the thing you need to remember. I said it earlier when I did the monohybrid cross. Now the dihybrid, we're going to make gametes. What are the possible gametes?
Big Y, little y.
And keep in mind that the possible gametes have one Y gene and one S gene.
That's always the dominant, right?
And well, no, because I mean, what are the possibilities? I mean, and we're going to get this. This was his actual law, the law of independent assortment, is that when we're making gametes,
so you can get Y's.
Any one of these two Y's could go with any one of these two S's.
Mm-hmm.
That's the independent aspect. They assort separately. The Y, which Y gene goes into a gamete has no effect on which S gene goes into the gamete. Does that make any sense?
So you could end up with a dominant
So
Y
let's
and
write
a
down these possible gametes from this
recessive.
one. The possible
Okay.
gametes from this one. Okay, the possible gametes. A big Y could go with a big S. Or the big Y could go with a little s. Or the little y could go with a big S. Or the little y could go with a little s. In other words, there are four possible gametes that this individual pea plant could make with respect to the Y genes and the S genes. Four different possible gametes. Because which Y goes with which S is just random. That's the independent aspect of it. Independent assortment. So there are four possible gametes that this person could make, this individual plant. Same thing with this one, exact same ones as it turns out. So it could be Y, big S, big Y, little s, little y, big S, little y, little s. And when we do the dihybrid cross, now keep in mind, we're doing a cross now between, we're talking about two traits. See, before we were just talking about maybe seed color. But now we're talking about seed color and seed shape. We're talking about two traits. And so now this is the dihybrid cross. This is a dihybrid cross. In order to get the hybrid, see this, they originally were start with crossing the true breeding plants. So these are the dihybrid. This is a dihybrid because it's got the big the dominant and the recessive. So again, if we use a Punnett square, now this is going to be how many possible gametes? Four here, so it's going to be a 4 by 4. 16 possible combinations.
All right.
So, if I write it out, I'm going to write it here. Let's see if we do this. I'm going to make it big enough so you can see it. And big enough so I can write in it. Let's see if I can do it. Let's see what we can do here. It's just easy if you can write all the things down. 1, 2, 3, a little further over this way. 4, a little bit. I'm making it big so you can see it. So there's my 4 by 4, but I left the top little open places to put my gametes. And it doesn't matter which one, if I put the ones that are the first person over here on the upper, that stuff doesn't matter. So, but I'll write them up there. Write up these possible gametes. And again, remember, your gametes will have one Y gene, one S gene. And there are 4 different possible combinations of what might be produced. And presumably this individual is going to produce millions or dozens, thousands of gametes. Some of them will be big Y, big S, some big Y, little s, etc. There should be of beta flows. That's what you expect. So let's write those up. We have a big Y and a big S, big Y, little s, little y, big S, little y, little s. And it's exact same as it turns out coming down. It's not always exact same depending upon what you're supposed to do, but in the one we're doing, they're exactly the same. Big Y, big S, big Y, little s, little y, big S, little y, oh. I didn't make this. I want to have 3 by 4. I need to go all the way down here. Sorry. Mistake. Paying attention. Okay, now it's 4 by 4, right? 1, 2, 3, 4. 1, 2, 3, 4. Yeah, 4 by 4. 16 possible combinations. I know it seems tedious, but we need to write them out. We need to write them out. Now, these are the gametes. What we're putting in the boxes are what the possible individuals, the actual genotype of the possible offspring. And they're going to have 2 Y's and 2 S's. Put the 2 Y's together, then put the 2 S's together. And when you write the 2 Y's, put the dominant first. When you write the 2 S's, put the dominant first. If there is one. Just for convenience. That just helps. So this one, there's both.
Y.
Maybe I'll use blue, maybe. Okay, and you've got to be careful. You've got to say it. Big Y. So 2 big Y's, 2 big S's. Now, let's just write it out. I know it's going to be tedious, but let's do it. These are 2 big Y's, a big S, a little s. There's a big Y, little y, big S, big S. Big Y, little y, big S, little s. I'm leaving room to write the genotype. That's the genotype, sorry. Phenotype underneath. The second box, the second going across. Big Y, big S, little s. Big Y, now 2 little s's. Big Y, little y.
Big S,
little s. Big Y, little y, 2 little s's. Coming down here. Big Y, little y. Big S, big S. Big Y, little y. Big S, little s. Oh my God, it's getting tiresome. Little y, big S, big S. Little y, big S, little s. You gotta be real careful when you're doing it to get the bigs and littles. I'll do the last one. Big Y, big S, little s. Big Y, little s, little s. Oh my God. Oh, these 2 little y's now. Little y, big S, little s. Little y, 2 little s. 2 little y's, 2 little s's. You see what I mean? How it's you have to be really careful when you're writing the letters, because some letters you have a tough time distinguishing your big letters from your little letters after writing a whole bunch of combinations. So be careful when you do it. 16 combinations. Some of them are the same, some are different, of course, but let's just see those are genotypes. Let's write the phenotypes, other than phenotypes in red. What would the phenotypes be? Let's see, what was it? Our yellow was the dominant, so it's yellow smooth. This means, we'll say yellow smooth,
Yellow smooth and yellow smooth.
Yellow smooth. Oh, that was pretty easy. But it won't stay that way, really? No. Yellow smooth. Those are all the same. In terms of phenotype, different genotypes. Again, same thing. Yellow, smooth. Hey, here's a different one.
Yellow wrinkled.
Yellow wrinkled. I can't spell. Yellow smooth again.
And yellow wrinkled.
Another yellow wrinkled. And then we have yellow smooth again. Obviously, most of them are going to be the 2 dominants.
Yes.
Makes sense, huh? Yellow smooth. Yellow smooth. Hey, here's a green smooth. here's a
Another
green
green
smooth again.
smooth.
Got yellow smooth. Yellow down. My God, let's write it down.
Yellow
Here's a
wrinkle.
yellow wrinkle. There is a green smooth and then a green wrinkle. Only one green wrinkle out of all those. But that's the double recessive. So, see if you get all those down. If you can get them down, please do. Then you can look at them. I mean, you can see this in books as they'll do the same thing with whatever traits they're using. It doesn't really matter. It's the same idea, but it's really useful to do one of these, more than once actually, just because then you get to see it, and it makes more sense. So let's put down the phenotypic ratios.
So, 15-1.
So we have yellow smooth. We can have yellow wrinkled. We can have green smooth and green wrinkled. Well, we already saw there's only one green wrinkle, so that was one of those guys. Let's count the yellow smooths.
11.
1, 2, 3, 4, 5, 6, 7, 8.
Oh, it is 9. I counted too wrong.
9. Yeah, better be. I would have goofed somewhere if it hadn't been. Okay, so 9 yellow smooth. Yellow wrinkled, there's 1, 2. Where's the third one? Here. There's 1, 2, 3. Yellow wrinkled, so that's 3. Green smooth should be 1, 2, 3. Yeah. And then 1 green wrinkle. So it comes at 9 to 3 to 3 to 1. Does it make sense what we did?
Yes.
Again, I want to stress when you're doing these, write down the genotypes of whatever it is, whatever they give you. But when you make the gametes, if you're doing a dihybrid, only one of the Y genes could be in any one gamete. Only one of the S genes. In this case, you could have a big Y going to big S or a big Y going to little s. That's what law of independent assortment says. It says when you have two separate traits, okay, the genes for those traits go into the gametes separately. They don't. In other words, what Y gene goes into a gamete has no effect on what S gene goes into a gamete. So for two traits, we could write this down to make it sort of easy. For two traits, the genes for these two traits, are independent of each other when forming gametes. I just stated it this way. You can state it several other ways, but all the same thing. So they assort independently. They move to gametes independently. The Y gene has no effect on the S gene when you form gametes. Which Y gene goes to a gamete has no effect on which S gene goes. They are independent of each other. Now Mendel, keep in mind, Mendel studied these pea plants. They have he studied 7 characteristics, 7 traits, like pod color and pod shape and all that. and there are 7 pairs of chromosomes. And the genes for each trait were on separate chromosomes. So in other words, the gene for this is seed color, sorry. The gene for seed color was on one chromosome. The gene for seed shape was on a separate chromosome. So they do assort independently. If they happened to be on the same chromosome, he wouldn't have gotten such a neat ratio. And it's possible that he studied some of the traits that were on the same chromosome. The numbers didn't make any sense. And it's very possible that happened, and he said, I can't make sense out of it. So he said, I'm not going to deal with it. But these made sense, and I doubt that he just by chance chose 7 traits on separate chromosomes. That would have been pure luck. I think he did just say, I can't make sense of these, I won't deal with these others. But these make sense. And by making sense, you can come up with some ideas that make sense. Then you can go on and discover other things like genes on the same chromosome. Anyway, that's the basic idea of Mendelian genetics. And as I said, you get the basic stuff, then you can start building on it, and building on it, and building on it. Okay, so let's talk about some variations on Mendel's laws as an example. Okay, let's see. So one variation I didn't put there, but I should have put linked genes. So we'll just say variations. Variations on Mendel's laws. And keep in mind, back in his day, in fact, it wasn't even until it took 100 years before they really understood what genes were and DNA and all this and chromosomes, all the details. So, but let's take linked genes. Let's talk about those linked genes. And those are genes for, we'll say, 2 traits are on the same chromosome. So if you're looking at 2 different traits, whatever they might be, and the genes for those traits are on the same chromosome, then they'll always go together. See, instead of segregating or assorting independently, they would assort together because they're on the same chromosome. Most of the time. Most of the time. Because what do you know about from last week and doing meiosis? What do you know happens during meiosis to give us a lot of variability?
Crossovers.
Crossing over. So because of crossing over, you can get genes that are on the same chromosome that can get switched from mom's chromosome to dad's chromosome by crossing over when you're making gametes. So if genes for the 2 traits are on the same chromosome, then they usually will appear in the same gamete. Okay, I'll put here. Will usually occur in the same gamete. Unless crossing over occurs. Crossing over occurs to separate them. Crossing over occurs so this is something can happen. And by the way, the further apart they are in the chromosome, the more likely they are to be separated by crossing over. Will usually occur in the same gamete unless crossing over. Yes.
How many gametes are possible from just crossing over?
Oh my God! Then we're getting so many unlimited numbers, right? Who knows? There's no set number.
But there is a calculatable number considering the number of. I know it's big, but I deal with big numbers.
Yeah. No, but the point being that you don't know how many crossing over events are going to occur with any one tetrad.
You don't know how many will occur. Okay.
And always they say you get at least one crossing over event, but you could have dozens within one chromosome, a whole bunch of crossing over. So it's totally random, apparently.
So just a large number, because that's a question from the
I
last
know.
lab.
I'd say unlimited number. Who knows?
Not infinite, but large.
Who knows? I mean, it's very large.
Right.
And the more chromosomes you have, the larger. I mean, so yeah, I mean,
I asked Colette, she said.
say again.
I asked Colette, she said.
Yeah, and I would
infinite.
say infinite, say uncountable, say unlimited.
Yeah, she told me.
Vast. Yeah, she said it was vast.
Yeah.
She said, "Stay away from the term infinite."
Oh, I know,
Yeah.
because obviously that's and just say
That's why I brought it up,
a
though.
huge number. I mean, obviously it's not infinite. An infinite's not a number anyway; it's a concept. But that's
Yeah,
okay.
yeah, right.
All right. What about things like what's called incomplete dominance? What's that referring to? Well, incomplete dominance. In some cases of flowers, certain flowers, you have a red flower and a white flower, and you cross those two. I mean, it's, red and white flowers are the same kind. You cross them, they'll all be red flowers because red's dominant. But other flowers, if you have a red flower and you cross it with a white flower, you get pink. You get something. Okay, so it depends upon the flower. Okay, so if you have, so let's just say if a red flower cross white flower, you get all red flowers. Then this follows Mendel's law. Okay. Well, but if you have and this does happen with certain kinds of plants, if you have a red flower and you cross it with a white flower, but if a red flower cross a white flower, gives pink flowers, so something in between, then this is incomplete dominance. Now, what's happening here?
That is happening.
And it's getting to what we mean by dominance, and it does vary with the gene. As I said, the simplest way to think about what's dominant versus what's recessive is the dominant gene says to do something, to make something, and the recessive says don't make something. So that would be the case. That would be the case in this one. In this one, if you have the red gene says to make a red pigment, the white gene doesn't say to do anything. So you get red flowers. Okay, but in the case of something like the one below, if you have a red gene, that might say make some red pigment, and the white gene might say to make some white pigment. But then if you have a plant that has both genes, those two pigments will blend in terms of their color and come out pink.
But doesn't that
So it's a blending.
imply dual dominance rather than incomplete?
Yeah, you could say that. Or and there's a whole bunch of other terms that talk about penetrance, how much the gene will be expressed. But I don't want to get into all that.
Who's the
But
bigger guy that can beat up the other
in
guy?
that case, it would be like codominance, which is the next one I was going to talk about. So
Okay.
you could say this was codominance. You could put it under the same category.
Okay.
Codominance. I'll give you the example. Codominance when more than one allele was dominant. And we'll actually talk about this next lab. I think we're going to get some human genetics. When more than one allele was dominant. The example I'll give is in blood typing. In humans, we call it the A, B, O blood type. Blood types. Okay, A, B, O blood types.
O is dominant.
A and B are both codominant over O, which is recessive. A and B are both dominant or codominant. We'll say they're codominant. O is recessive. So if I go give you, let's take phenotype and genotype. If you're type A, by the way, you only get one A gene or one B gene, or I mean, you have two genes for your blood type. Could be an A, a B, or an O, any combination of those. So type A, you could have, I'm going to write it this way, A or A O. If you have two A genes, one from each parent, you're type A. If you have an A gene from one parent, an O gene from the other, O's recessive, doesn't say anything, you're type A. Okay. If you're type B, B, big B, or big B, O. What about type AB? Type AB, we've got an A gene from one parent and a B gene from the other. A says to make this A protein on your red blood cells. B says to make this B protein on your red blood cells. You're type AB. In order to be type O, you have to have two O genes. So that's the idea. We're almost done here. Just a bit. But anyway, so the point is that both A and B, if you have an A gene, it says make the A protein. If you have a B gene, it says make the B protein. So if you inherit both, you're going to be type AB. Oddly, the ratios are real funny. In, like, say, a European population, type A is like 40-some-odd% of people. Type O is 40-some-odd%. Type AB is maybe 7%. Type B maybe 11% or something like that. But in the Asian population, the A's and B's are reversed. Type B is more common. Type A is more common. Who knows why? Nobody knows why. Yeah.
Do you know about the RH protein?
I do.
Which is the dominant?
Positive. Because the D, the RH positive, which is the D protein, the D says make that protein, you make it. And the little d, which is the recessive, says don't make it. So if you have a big D and a little d, you're still positive. In the European population, about 85% are RH positive, about 15% RH negative. In the African American or the African population, about 99% RH positive and 1% RH negative. Who knows what? Wait a minute, I still have a couple more 5 minutes, don't I?
Yeah.
Let me just tell you a couple more things real quickly. Oh my God. Some other things. Okay. Another thing. Polygenic inheritance. So let's talk about that real quickly. These are kind of important ideas, which I just want to at least throw out here to you. Polygenic inheritance. You know, if you're talking about something like height, there's not one gene for height. There's going to be a whole bunch of genes involved in height. So that would be polygenic. Polygenic, we'll say polygenic inheritance. Many genes involved. We're just looking at, one pair of genes, but there'd be many genes involved, something like height or whatever else. And that's why in something like height, there's a huge variation from very short to very tall to most people somewhere in between, because there's a whole bunch of genes involved, plus of course environmental factors. Another thing is pleiotropy. That's a good word. Pleiotropy. That means that one gene can have many effects. One gene can have many effects.
I was actually reading about the gene for your earwax is also the same gene that controls for how much body odor you have.
Oh, let's talk about body odor. No, I don't want to talk about body odor. Sure. Yeah, is that right? I didn't know that.
Apparently, there's a gene for dry earwax that is mainly in Asia, and it's also one that controls for like a lighter, like a less potent body odor.
No, by the way, body odor, you don't have a natural body odor. The body odor deals with the bacteria working on your excretions. Mainly, we're talking sweat. So, but yeah, okay, I can imagine it could have an effect on some of the secretions. Yeah. And that can then have an effect on bacteria working on it and the odors that are emitted. But we don't want to talk about body odor. Oh, we could talk about it. But anyway, back to pleiotropy. We've been dealing, and Mendel dealt with, fortunately, to have made sense out of it, one gene, having an effect on one trait, like color of the seeds of the plants. So we could say that for eye color, we said there's more than one gene involved. So that may be polygenic, it might be three or four pairs of genes that might be involved. Who knows, 2 or 3 pairs. But with pleiotropy, when one gene can have many effects, we were dealing with, one gene, one enzyme. Remember we were talking about that, or one gene, one protein. Well, okay, but in some cases, the enzymes involved, maybe as an enzyme is being made, or maybe some other kind of, maybe a control protein of some kind, but it might be some kind of a switch protein or switch enzyme. In other words, if your gene makes whatever the product is, it'll switch on a whole bunch of other genes to start functioning. Whereas if you don't have that protein, it won't switch it on. So that way, one gene making one protein could have a big effect depending upon whether it switches on several more genes or doesn't switch on those other genes. That makes sense? Mm-hmm. And that's how, so not everything is simple that, hey, here's a gene for eye color, here's a gene for seed color, or something like that. And in some cases, it might be some kind of a control, a gene that has some protein that has control. Anyway, so things are obviously, not as simple as Mendel originally said. But again, the importance of Mendel was to make it understandable, so then we could ask more complicated questions. All right, so we did it. So the exam will go through today, and I'll put on my CR some sample questions, okay? And then on Wednesday we'll have new material. It won't be an exam, but on Friday we'll do reviews.
All right.
All right.